Good afternoon. Thank you for inviting me to present my work here. I'm Kyra Hamilton, and I study my PhD with UCD Dublin and conduct my research between Teagasc in Ireland and ag research in New Zealand. Today I'm going to focus on the paper, Survival of the Fittest, Ecological Fitness in Teladorsagia circumcincta of known anthelmintic resistance status. Now, the background for this project lies with nematode parasites of sheep, primarily with Teladorsagia circumcincta. I'm sure you're all aware of the importance of parasitism as a production limiting factor for grazing livestock. And most of you will also be aware that the long-term reliance on anthelmintic drugs to control these worms has resulted in the escalating issue of resistance among the worm populations. Now, a great deal of money and effort has been invested in finding ways to slow the development of anthelmintic resistance to these important drugs. And this is essential to ensure the sustainability of our industry. Now, several years ago, something really interesting and perhaps a little surprising happened in some on-farm studies conducted in New Zealand. So what you can see in this graph here is that when a complex of resistance management strategies were applied to a series of commercial sheep farms, all of which had pre-existing issues with anthelmintic resistance. And over the course of 5 years of monitoring, there was an overall improvement in the treatment efficacy against Teladorsagia circumcincta. Now, this occurred in all 3 of the older anthelmintic classes, so our classic BZ, levamisole, and macrocyclic lactones. So this observation raises a question as to the possibility of managing our way out of a resistance problem once it has developed. So my PhD is studying and aiming to try and understand one possible mechanism for this to occur. So what we're going to look at is fitness. So the hypothesis we came up with was, are resistant mutations resulting in ecological fitness costs in Teladorsagia circumcincta? Now, fitness is defined as the ability to survive and reproduce. In internal parasites, it therefore involves establishment in the host, reproductive ability, infectivity, and lifespan. So developing in vitro assays to quantitatively measure aspects of parasite fitness required assessing traits deemed essential for the life cycle. So for Teladorsagia circumcincta, this included measuring fecundity, migration after exposure to hot temperatures, egg hatch percentage after exposure to cold temperatures, egg hatch under regulated temperature, and exchievement from free-living to parasitic stage 3 larvae. Now, 4 isolates of known anthelmintic resistant status were used. So isolates D and M were multi-drug resistant, and to save confusion here, I'm going to refer to them as R1 and R2. And then we also have isolates T and W, which were pan-susceptible. And again, to save confusion, I'm going to refer to them as S1 and S2. So R1 and R2, resistant 1 and resistant 2, and S1 and S2, susceptible 1 and susceptible 2, as you can see from my table here. So in total, 8 lambs were infected, 2 for each isolate, and they generated material for the in vitro fitness assays. A larval migration inhibition assay was used to confirm the resistant status of the isolates against ivermectin. And from this, R1 and R2 were confirmed resistant, and S1 and S2 confirmed drug susceptible. Now, there were 5 assays selected to quantify aspects of parasite fitness. So we used larval length as a measure of fecundity, but we also know from other research that multi-drug resistant Teladorsagia circumcincta are shorter than their susceptible counterparts. An egg hatch assay at regulated temperature was used to measure the basic hatching ability of the isolates. We also looked at the percentage exchievement from 3rd stage free-living to parasitic larvae, and we did this using the exchievement assay. And after this, we measured the migration ability of 3rd stage larvae, and this involved exposure to hot temperatures for a set length of time, and this was called the accelerated aging test. And then finally, the cold stress test measured the percentage development of egg to 3rd stage larvae after exposure to cold temperatures for a certain number of days. But what were the results? So after initial analysis, the assays turned up some interesting results. the larval length assay assessed the lengths of 100 different larvae for each isolate and revealed that S2, so the susceptible to isolate, was found to be significantly longer than all other isolates. The egg hatch assay revealed that isolate R2 had a consistently higher hatching percentage than S2. So in this table here, all the values with the same superscript had no significant differences. If there's a different superscript, there's a significant difference. And then we followed on with the exheathment assay. So for the exheathment assay, larvae were incubated in artificial rumen fluid for up to 24 hours before the percentage exheathment was determined. Now, from our results, and in the box I've highlighted in red here, this is where our significant results occurred. Isolates R1 and S1 had a higher percentage exheathment than that of isolate S2 after 24 hours. For the cold stress test, similarly, I've plotted a box plot. So the fecal samples here were incubated at 4 degrees Celsius for various lengths of time. And after this, the percentage development was determined and the initial time point was used as a base level. So time point 0 was used as 100% for the hatching percentage. Again, here I've highlighted in red the most significant area where the results were found. The area with the most significant results were found. So at time point of 4 days, for the cold stress test, this revealed differences between isolates R1 and R2 and R2 and S1 in their egg to stage 3 larvae percentages. Again, the accelerated aging test. So interestingly here, we looked at 3rd stage larvae migrating after incubation at 30 degrees Celsius for various lengths of time. So as you can see, we have hours extending from 0 to 504 hours of heat exposure. So the results of the migration following the accelerated aging assay showed that all the isolates displayed a reduction in their migration ability over time. In particular, the only significant differences were seen between isolates R2 and S1 and isolates R2 and S2 at time point 504 hours of heat exposure. Again, I've highlighted it with a red box because there's a lot of information happening here. But what is so interesting about these results? So these results don't specifically give us a definitive answer as to whether or not resistance to one or multiple drugs results in a fitness cost. But what it does show is that there are fundamental differences between the base and or background level fitness between the different isolates. Apologies, I'm just going to go back a slide. Yeah, so fitness doesn't necessarily correlate with resistance status, but with the wide variation in background fitness, it does suggest that in order to prove the hypothesis or disprove the hypothesis, we need to do more research, which brings us on to my current studies. So currently, there are studies ongoing to investigate the relationship between resistant genotypes and fitness in a single population. So for this, I have used a single isolate population and split it into its different subset known multi-drug, and we've done this using multi-drug resistant populations that we know are multi-drug resistant. And in order to split the population, we have used a different combination of anthelmintic drugs. So we've used unselected single, double, and triple combinations for this. And through using this single isolate population, so we hope to reduce the ecotypic variation as happens when you're comparing multiple different isolates from different sources. And also we hope to minimize the background variation in fitness as we've seen from this current trial that I'm doing. So in summary, there are a large variation in the background fitness of different isolates. And the hypothesis that I have worked on cannot be conclusively accepted or rejected from this trial work. However, that's why further testing is underway, and this work will hopefully work towards helping me being able to conclusively accept or reject the hypothesis of the work. Thank you.
Thank you very much, Kyra. Very interesting. Okay, I don't see any question from the Mentimeter, but maybe I have one. So those isolates were from sheep, isn't it?
Yes, so my work is in sheep parasites.
And do you think that this kind of ecological experiment can be applied also to other gastrointestinal nematodes, or are you planning to do other studies?
Currently, my studies are only in sheep, but if we look at the specific life cycles of other parasites, we could adapt it similarly towards them. I think it's important to look at aspects that are essential for their life cycle. Like, you know, with liver fluke, you have specifics towards them where they migrate into the liver, and the most damage is caused during the juvenile stage, during their migration.
Oh, there is a question. Okay. Have you considered collecting early and late HX and then testing whether the larvae have the benzimidazole SNPs?
SNPs, single nucleotide polymorphisms.
Yeah.
I don't do the molecular work, but so I don't know specifically about that, but it would be interesting in the future to look at collecting between early and late in the year so that we could compare the fitness then, because that's part of the work I'm looking at with the cold stress test and the accelerated aging test. It puts them under heat and cold stress to try and emulate what would happen if they were put under such stresses at different times of the year in a fake environment in the lab.
Okay, thank you very much, Kyra. Thank you.